finished with this, we will upload it to our YouTube channel. So if you'd like to view this later, you can certainly do that. And today's program will be about 45 minutes to maybe an hour, somewhere around there. Okay, well, let's go ahead and get started. So today's guest is Dr. Valerie Paul. She is the director at the Smithsonian Marine Station, also located in Fort Pierce. And today she's going to be talking about a devastating coral disease that's termed stony coral tissue loss disease that's impacted Florida's coral reefs since 2014 and has now affected all reefs except the Dry Tortugas. This disease kills over 20 species of reef-building corals, and researchers at the Smithsonian Marine Station have been studying this disease since 2017. And results of their studies on the ecology, the search for causative microorganisms, and the development of treatments in the form of probiotics will be presented at today's program. Jamil will also be putting the link to Dr. Paul's personal page from the Smithsonian Marine Station in the chat, so you can learn a little bit more about her if you'd like as well. So Val, I am going to turn it over to you. It's all yours. And I'll be popping back in to ask questions every now and then.
Okay, Erin, thank you. Let's see, I think I need to share my screen with everyone. So let's go ahead and do that. Let's see here. Okay, I got it. Share. Everybody see that? Let me go ahead and put it in presentation mode. Welcome everyone. It's great to see participants from all over the country, it sounds like. So that's really wonderful. Really glad that you could join us this morning. Can everyone see my screen and hear me okay? Erin, you might have to give me a nod. Somebody raised their hand already too, so I'm not quite sure.
Yeah, it looks like everybody's able to see you and hear you. If anybody's not, please just let us know in the chat.
Yeah, there is a hand raised, so I'm not quite sure. You might have to look into that, Erin. So anyway, good morning, everyone. And I want to tell you a little bit about our work here at the Smithsonian Marine Station on this pretty bad coral disease that I'll tell you quite a bit about today. And I really want to thank Erin and everyone for inviting me to present this work. We've been working, as Erin mentioned, on this for the last a solid 3 years. And we keep coming up with more questions than answers, which is often typical in science, but I'll certainly tell you a little bit about what we have learned. We've been able to publish some of this work, so if anyone's interested, just let me know. And it's just been a big focus of our laboratory. As Erin mentioned, I am the director here at the Smithsonian Marine Station, so a lot of this work today is done by a team of researchers and clearly not just myself. So I'm really going to focus on acknowledging them as well. So let's go ahead and Erin told you a little bit about stony coral tissue loss disease already. It seems to be an infectious disease. It can be transmitted by corals touching one another or through just the water itself. Diseased corals in a bucket, for instance, that water can transmit disease to other corals, and over 20 coral species are affected. A lot of the boulder corals, the big Orbicella, Montastrea. So far this disease is only known in the Caribbean, but it can affect almost 25 species of Caribbean corals. The mortality rate can be very high. I'll show you a few pictures. And the distribution throughout the Caribbean seems to keep spreading, and I'll show you that as well. So these are just some pictures of what this disease can look like. So unlike bleaching, where the coral animal is still alive and they just lose some of their coloration because of the symbiotic algae that live in the corals, these are actual tissue loss. And so the coral is dying as the disease progresses. And so what you see here are 3 different species of corals, Montastrea cavernosa, Dichocynea, and brain coral. I think that's Pseudoultramonis. And the brown coloration on these colonies is the nice healthy tissue. It's loaded with these microalgae called zooxanthellae, they're within the group Symbiodiniaceae. They're dinoflagellates. And they give the coral its nice healthy color. They're photosynthesizing, so they can help, bring nutrition to the coral animal. And they're just a really important part of the ecology of the animals. And so as they start to die in lice and the coral starts to die and die away, it just leaves the white skeleton behind. And so that's the white you're seeing here. They're not bleached coral polyps, it's actually dead coral, and it's just the white skeleton. And the skeleton can get overgrown pretty fast by other things like, algae mostly and things like that, biofilm and algae. So it'll start to discolor pretty quickly. So the fact that so much of this skeleton is white here is telling you that the disease is progressing really quickly across the colony, faster than the algal growth can cover up that dead skeleton. So it can move quite quickly. And this just shows you the distribution. Erin mentioned this already. When we started working on this, it was 2017, and it had already started progressing away from the center of where it was first started. So on these maps, on the left you see 2014, on the right 2020, but the red area is where the disease is prevalent. And you can see now by 2020 that really it's just the, and by 2021, it's really just the Dry Tortugas that remains disease-free in Florida. And at the same time, it has started moving around the Caribbean, which is very concerning. As you heard, it started in Florida in 2014, we think, and then started popping up in other Caribbean nations, somewhat, I won't say randomly, but not in a real predictable pattern, if you will. You can see how it's opposite ends of the Caribbean region here in 2018 in Mexico, and in the Windward Islands there. And in Mexico, it progressed all the way down to Yucatan within about 9 months and caused quite a bit of devastation. It's now found in both Belize and Honduras as well. Puerto Rico's reporting it, US Virgin Islands, Dominican Republic, and other areas. So it's just sort of popping up around the Caribbean. And this is what kind of damage it can cause. Again, remember that the white on these colonies is the dead skeleton showing up as the coral tissue dies. And you can see from February, this is about a 6-week time period from February to mid-March in 2019. This disease was spreading through the US Virgin Islands at that time. And this is courtesy of Sonora Myling there, who provided me this time series of a very large boulder brain coral that completely succumbed to the disease in this time period. And so it's really devastating as a diver to watch this and to watch these corals die. And so here's a picture of a reef in, a video actually, of a reef in the Florida Keys that we were working on in 2018. And this video is courtesy of some of our colleagues at the Florida Fish and Wildlife Research Institute down in Marathon. We were diving with them at that time. So you can see those blotchy white patches on some of these corals. And then you can see like that one coral we just went over was almost completely healthy. All the rest of these are just boulders left behind from the corals dying. Sorry, this is a little dizzying. There's some other ones that are partially dead. So the surprising thing here is that you can find corals on the reef that seem unscathed. So there's definitely, even within species that can get the disease. So there's definitely some differing susceptibility. And of course, we know that with any disease, right? Like with COVID-19 now, we know that different people have different susceptibilities, and some can get much sicker than others with the disease. So it's somewhat similar here for the corals. Although we have no idea, as I'll tell you as we move along, that we have no idea what's actually causing this particular coral disease. So this is just some of the approaches that we're taking in my lab to move this research forward, and with a large team of investigators. We're looking at some culture independent analyses at the microbial communities, and Julie Meyer, who's at the University of Florida, has been our partner for this work. We've been collaborating together on coral disease work for, oh gosh, almost a decade now. We first started working on a different disease called black band disease, but we have since switched a lot of our attention to this disease because it is so devastating and still spreading. So this is microbiome analyses that we've published. This is in Frontiers in Microbiology, if anyone's interested. We've got some culture dependent work where we're actually culturing the bacteria off these coral colonies, and you'll see a lot more of some of that work later because it is partially leading us to some of this work we're doing for treatment of the disease, and that is trying to develop some probiotics for the disease. So this is some of the team. I mentioned Julie, she's there on the right. And I also want to point out, I hope you can see my cursor. This is Blake Ushijima, who was a postdoctoral researcher here for about a little over 2 years, working with us on this disease. He's now a faculty member at the University of North Carolina at Wilmington. And Dr. Greta Abbey, I lost my cursor. Here in the black shirt in the middle is a very well-known disease researcher from the University of Hawaii who has worked extensively on coral diseases in the Pacific, and she was really helpful in getting us started working on this disease here. We started off, and Blake was very interested in trying to see if he could find some of the pathogens that might be causing this disease, and we suspected bacterial pathogens could be involved because they are in other coral tissue loss diseases, but we otherwise, of course, have no idea. And so he isolated lots of bacteria, hundreds of bacteria from the diseased corals, and then got them in pure culture and retested them, usually in pools of samples, so 5 at a time. And that's what you're seeing in that middle row there. Here's my cursor. The middle row here shows you testing 5 at a time. And then these are actually tested on little small pieces of coral, fragments of coral. And then if there's any tissue loss, then we know we might have a suspect bacteria. And that you see in the bottom row with an unhappy face on the coral. We never got anything from these preliminary first efforts that was consistently causing tissue loss. So we do not believe we have anything that consistently led to coral disease. But we did isolate a few bacteria, including some Vibrio coraliticus strains, which I'll show you in a minute. And some of those can sometimes cause tissue loss. So we actually have some strains that we use for testing our probiotics that are that can cause tissue loss in some cases, but not others. And certainly we don't believe that they are the causative organisms for stony coral tissue loss disease. One thing we found out early on, and this is published, Greta has published one paper in Frontiers in Marine Science that goes over a lot of our first work on this disease. We found that treating them with antibiotics could halt the disease. So here you see the kind of little coral fragments we're working with. They're a couple centimeters on the side. And a control, which was just untreated, progressed pretty rapidly. And by day 10 here, or day 4, the disease has completely wiped out this little coral fragment. You just don't see anything left. You just start to see some spongy algae growing back over the coral skeleton. Whereas for the corals that we treated with a mixture of amoxicillin and kanamycin, each at 50 micrograms per mil in the aquaria, we pretty much stopped the disease. And you can see it didn't really progress much from over that same time period. So bacteria are implicated. We don't know if they're the cause, but they're certainly playing some role. And we also started seeing in our isolations, as well as some of the field work we were doing, this known coral pathogen, which is also a shellfish pathogen in the oyster culture, aquaculture business. This one can cause vibriosis, which can wipe out some of the larvae. So Vibrio coraliticus is a well known for causing tissue loss in a variety of different invertebrates. A little assay test was developed by one of our collaborators which is almost like, a pregnancy test, if you will, or something, where it's just a colorimetric test. You have a control line. Basically, you spot a little bit of tissue, a little dabble of tissue of the coral that you want to test. And then it runs up the test strip, and the sample's moving by this control line. And then if the test line shows up, that means a positive test. Okay, so a negative test would look like this, one stripe, positive test like this. And this was not developed for stony coral tissue loss disease. This assay was developed for the shellfish industry, which of course, has value in detecting the presence of this pathogen that can cause shellfish damage. So we were able to use the assay to look for Vibrio coraliticus in the corals. And it's present in about 20% of the lesions of Monstera cavernosa that we looked at. And that was whether we were looking in northern part of the Florida reef system or down in the Florida Keys. It seemed to be about 20–25%. And so why does this matter? Well, the corals that are co-infected or have a secondary infection with Vibrio coraliticus die a lot quicker. You'll see some more of these kinds of graphs as my talk goes on. And this is the percent surviving coral fragment. So 100% means nothing died, right? They're 100% are surviving. But you can see quickly that the ones that are positive for Vibrio coraliticus toxin, that the test is actually detecting the toxin, not the bacterial strain itself, but the toxin it produces. This Vibrio toxin that can lyse the coral tissues died much more rapidly. This is just 21 days, and all of our test corals, 12 here, were dead in that time period, and none of the ones that didn't have the that didn't test positive for this toxin. So it's playing a role in disease progression. We do not believe it's the primary cause of the disease. And as I mentioned earlier, we really don't know what the primary cause is. So we've started to turn our attention more to treatments. And of course, the state of Florida was very interested in treatments, as are other Caribbean nations, as this disease is moving around. And everybody's grappling with the best way to treat these. So people have tried a number of things. One is, of course, just colony removal, which is certainly not very practical. Some of these colonies are really large, and then just handling them and everything could spread the disease around more. It's not a practical thing. But treating with chlorine powder and sort of these marine epoxies was tried. It was not terribly effective in the test cases in Florida. So what people have been doing in the Keys and up in the Fort Lauderdale, Miami region is using amoxicillin, an antibiotic, in a proprietary paste made by colleagues called CoreRx Paste. And it's mixed together and spread over the area of the lesion where the live tissue closest to where the tissue loss is occurring. And so here it's like a paste applied underwater, and it seems to be pretty effective. Sometimes they do need retreatment, sometimes fairly frequently, but it does seem to help and has probably saved many corals in Florida because the treatments have been ongoing in a kind of a trial manner for a couple of years. We started looking at a different approach, and that was using some of these bacterial strains that we had accrued and seeing if they could be applied in a positive way as probiotics. So probiotics are live microorganisms that confer a health benefit to the host, right? And we always hear about probiotics for humans. Some of these products are advertised for their probiotic potential. In humans, it's usually for your gut health, which, as we're learning, I think all you have to do is read the headlines in the newspapers every day. We're learning more and more about how the microbes in our guts can affect us from everything from our mood to depression and anxiety to, all kinds of diseases that seem unrelated, like our immune system and cardiac function and everything else can be related to our microbiome constitution and our microbiome health. So you know, some of these products, of course, have come out, but it's also used a lot, of course, in aquaculture and even in agriculture in some cases. So we've been applying this approach, and what we're looking for are things, bacteria, live strains, of course, that are not harmful to the corals. We've been looking for things that produce antibiotics. Here you see some bacteria on these auger plates that have a zone of inhibition around them, an area where this other test strain can't grow or doesn't grow. And that usually is indicative that these bacteria here are producing something, some kind of compound usually, like an antibiotic, that is inhibiting this growth. So we do a lot of these kinds of assays to look for it. We're looking for things that aren't already used in human and veterinary, as human and veterinary probiotics, and that can grow quickly and don't require a complicated media to keep the cost down. So in our earliest testing, we actually got lucky and came across one, this Pseudoultramonis strain that we call MCH17. And you can see it here. It's kind of this yellowish color, and it produces a nice zone of inhibition around it. That's the strain. And this is, we were able, in my laboratory, we do a lot of chemistry and chemical ecology, and we were able to determine that this known antimicrobial compound called Coromycin was responsible for most of this antimicrobial activity that we're seeing on the plate. Again, this zone of inhibition showing these bacteria on the test strains don't grow. But it also produces, we studied this strain very carefully. My colleague Julie Meyer has done the complete genome of it. And so we also know that it's producing some of these other compounds, and we've confirmed marinacine production in the genome, but tetrabromoperol genes are also in the genome, as well as we can confirm that it's actually producing that small molecule, which is a very interesting compound. This is work I won't talk much about, but Jennifer Sneed might mention in a few weeks. We've been using TBP also to stimulate coral larval settlements. So some of these probiotics may have multiple positive roles for their application. This is a little complicated, but I just wanted to let you know that the mechanism, how Coromycin produces its antibacterial activity, is known. This compound is actually studied a fair bit in Vibrio cholera research. So it's known to be very active against Vibrios, which is interesting since we know those Vibrio coraliticus strains can be important. And it's a sodium pump inhibitor. So it can definitely affect the biochemistry of the bacteria and cause them to not be able to function properly. So what about if we treat corals with this strain, MCH17? So here we have two pieces of the same coral. So we just take the piece of coral collected in the field, all of course under permit and everything, cut it in half, and then treat half of it with our strain MCH17, which we're just adding into the seawater in the aquaria, and then not treating half. And you can see there's quite a big difference here, right? After almost 11 days to almost 2 weeks, the control piece, the untreated piece, has died, just like I showed you before, whereas we've halted the disease, in this case, on the treated piece. And so this was really exciting. We've got quite a bit of data now on this, 22 corals we've tested this way. You know, not every piece gets halted completely like that picture I showed you, but again, if we look at these survival curves, you can see that the ones treated with MCH17 here in orange are surviving much better, meaning they're dying less than the control pieces. And so the strain seems very promising. This is all very statistically significant. And these graphs are looking at the disease progression rates from graphs, and we estimate the area under the curve, not these survival curves, but some other disease progression curves. And we see the exact same thing, that the ones treated with MCH17, which has a higher area under the curve, means that the disease is progressing a lot less. So we've done a bunch of other things. We've done safety testing with the strain on a bunch of other different coral species, just to make sure that if we take it to the next level and actually try to take it out into the field, do we see any signs of stress on other species. Again, this was a bacterial strain isolated from Montastria cavernosa. So most of our testing has been on Montastria cavernosa, but we want to make sure it doesn't cause harm to any other coral species. So we were able late last year to take, actually early last year, sorry, it's already 2021, but about a year ago, we were able to start some field trials for the first time. We believe this is the first field trials of probiotics on a coral reef anywhere in the world. And we've been tracking these very carefully. Colonies have been monitored over time, and then we're using a lot of other analyses to try and assess, how much we're impacting the coral or impacting its native bacterial flora through understanding the microbiome and other things. And also to try and see if we can detect the strain actually incorporating itself into the coral, which has been actually challenging. We have not been able to detect it in large amounts, which is probably a good thing. We don't want it to overwhelm the native bacterial community either. So here's a few pictures. It's taken some perfecting, and I want to acknowledge Kelly Pitts, who's been a member of our lab for the last couple years working on this with us, and especially to acknowledge the team of our collaborators, Brian Walker at Nova Southeastern University, because for the last year, the Smithsonian has not been actively diving, and so they've been doing almost all of the field trials for us, and we really, really appreciate their efforts and their work because they've been fantastic. Did take some developing. Kelly was kind of instrumental in developing these bags that just look like giant trash bags that sit down on the bottom, and they're weighted down with lead line. And then injecting the MCH17 strain in seawater in a syringe into the bag, and then letting the corals go for about 2 hours. The cost is really cheap, so growing these up is relatively inexpensive. Of course, the field labor is a little bit more so, but let me show you what the video looks like of just kind of injecting this in. So this looks like there's a ton being injected here, right? But this is just right at the exit area of the syringe tube. And as the video backs off a little bit, you'll see the bag and everything, and you really can't see a lot of the material there. But these sit for about 2 hours on the reef. And then we're hoping in that period of time that some of the microbe will be incorporated into the coral, and that those can be retained by the coral for their beneficial properties. So we've also been developing this paste. Again, I want to acknowledge Kelly and another staff member, Yes Marie De La Flor, who have been fiddling around with this recipe of this PVP sodium alginate paste, also non-toxic, relatively cheap, and then the probiotics can be mixed into this paste. And the advantage of this is it's a little bit more like the antibiotic treatments, and it can be applied right directly to the lesion edge, and so that can help that really well. So then the challenge is, and I really have to acknowledge Kelly here because she's been doing most of these photo analyses from the field photos, trying to determine then, well, what are we seeing, right? The obvious question, is it working? I'm sure everybody's wondering that. And it's a little hard to tell sometimes because some of these corals move pretty slowly in terms of the disease progression. These are all Montastria cavernosa, so they can sometimes naturally fight off the disease pretty well on their own, unlike some of the more susceptible species of corals. And so determining how effective this is proving a little challenging, but I'll show you some results. Kelly first has been working around with kind of just classifying the disease severity from these photos, all the way from healing, which we have seen a few cases of, not progressing at all, which is also a positive thing, very slowly progressing or quickly progressing or dead, of course, which is what we're trying to avoid, both quickly progressing and dead. And so this would be one she kind of considered slowly progressing. And again, this kind of whitish area that you see around the coral skeleton is the area that we would consider the lesion, the area of the coral that is showing the most effects of stony coral tissue loss disease and where the tissue loss is occurring. And it's actually pretty hard to tell that this is even progressing, but I think she found a few areas where she thought it was, like maybe down here along the bottom or up here at the very top corner or whatever. Anyway, she called that one slowly progressing. There's the arrow indicating that one area. And then the quickly progressing are easier to tell, right? So you can see this poor coral is already just kind of fragments of tissue on a much larger boulder. It's already been impacted by the disease by the time we got to it. But you can see that this whole area over here is dying away. So this one was actually, and it's continuing to progress. So that one would be considered quickly progressing. A couple of areas where you can see that. So, and one thing that was really interesting that Kelly picked up is on this one coral that was treated with the probiotic bag, this this area looked pretty good, like it was starting to heal up, and then this area looked like it was still progressing. And she later found, by going back to this video that I will show you here, of the syringe contents being injected into the bag, that all the contents kind of went into this one corner of the bag and not to the far end of the bag, which is where this area that wasn't looking very good occurred. So you can see how all the probiotic pooled up over here and not over here. So we've learned we have to be very careful to make sure that the probiotics evenly distributed through the bag, because this was a case where this end of the coral looked pretty good and this end didn't. And apparently we sort of missed it with the syringe treatment. So here's some results, preliminary. This is still ongoing work. These are just the number of corals in each category, and BS2, you know, you might wonder what that stands for, but it just stands for Broward, because we're doing these in Broward County down by Fort Lauderdale, again, through our collaborations with Brian Walker and his team at Nova Southeastern University. And it's site number 2. We had an earlier site last year that we worked at that was called BS1. So here's, and this gets a little confusing because we have both the probiotic paste method and the control paste that we're comparing. And one good thing here is that there are many fewer quickly progressing than there are in the control if we treat them. So that's good. But I suppose one bad thing is we haven't completely reversed all the quickly progressing ones. There's still one that is. With the probiotic bag, we actually saw slightly better effects. We have some that were actually healing, a couple corals. Some are still slowly progressing compared to quickly progressing here. So that again seems a little bit positive, but relatively small numbers, and we're still perfecting methods and getting this going. What we hope to do in the future is set up a site down in the Florida Keys where there are a few more diseased corals and give that a try. So let me go through the rest of this. Oops, sorry, go through these quickly. So just to summarize, we have bags and paste methods. We hope to treat some of the corals in the future with both simultaneously, because it seems like each one sort of has its advantages and there's no reason not to just do both. And then we see some evidence that we're slowing down the disease and sometimes even healing the corals with our treatments. Clearly have more to do. And Kelly's hoping to do a little bit more 3D modeling of the corals to be able to more accurately quantify the differences in the treatments. And so analyzing these photos and trying to understand what kind of effects we're having is being very challenging. Meanwhile, we're continuing to develop other probiotics. We have a library of about 2,000 isolates from these different corals, and we're looking for zones of inhibition there. These are what some of the testing looks like. The arrows here kind of indicate a couple where it's a little hard to see, but you can see there's some zones of clearer zones around them. So we're going through, we're trying to characterize them with 16S gene sequencing to be able to say something about what the taxonomy of the bacterial strains are. And we've got quite a few that we're growing up for further chemical studies to be able to say a little bit more about what kind of antibiotic compounds they're producing. So there you can see the zones of inhibition a little bit better there. To start to study the chemistry, this looks pretty yucky, but we actually use these big old cake pans with agar, right? You grow these bacteria on agar plates and grow up the bacterial strains in larger amounts so that we can isolate some of the compounds that they're producing and figure out what the compounds are. So that's kind of fun. One quick example here is a strain we isolated from an Orbicella fabiolata, another big brain coral, but a different species here. We were able to characterize the compounds. This compound called thiomarinol is one that it's producing, and we tested it. So this is the laboratory work. It's actually the easy part. The testing on the corals is really challenging, but this one didn't seem to be any more effective on Orbicella fabiolata than, so here's our survival curves, here's the control, but it wasn't much more effective really than and not statistically more effective than just the MCH17 by itself. So we didn't see a lot of reason to move this one forward. We haven't been working a lot with it, but we are continuing to work with our strains and trying to find some other ones that might work as well or even better than MCH17, and also looking at possibly mixing some of these strains. A lot of the probiotics that we might take, like in the yogurts and things like that, are actually mixed strains of bacteria. So there's reason to think that, you know, possibly mixing them up could be extremely useful. So developing these multi-probiotic treatments is something on the future horizon as well. So again, I just want to acknowledge all the people that have contributed to this. Clearly, it's not my work alone. I've mentioned to you some of the key players, but there were a lot more. We had, up until the shutdowns from the pandemic, we had a number of interns working with us, and they were extremely helpful. And then whole teams of people working on, you know, either the microbiology or the chemistry, and all of our collaborators. So it was definitely a very big team effort. But also we've had a ton of help. We've had support from the Florida Department of Environmental Protection. Our first grant was from National Science Foundation in the form of one of their rapid grants, because we were really trying to get off the ground and understand a little bit more about what this disease was doing to our reefs here in Florida and now around the Caribbean. And then all of our partners that have helped us, many different people, but particularly for the field trials, Brian Walker, and that's Brian there, and some of his team. This was the very first group of people, and I early last year, before all the COVID shutdowns and everything, when we were able to get out and do some of our very first treatments. So that was exciting. And I'm open to questions, I think. So that's the end of my talk. I'm going to go ahead and stop sharing.
Okay, excellent. Well, we do have some really good questions here. I'm gonna apologize in advance if we don't get to all of them. But Val, we have had a couple of questions concerning the water temperature, if the disease survives in cold water or if it just seems to be a warm water environment, and if that has any correlation to the disease that you've found.
Yeah, there have been some nice papers on the epidemiology. Haven't seemed to implicate temperature as being completely, well, certainly we know it can spread in Florida. You know, the range of temperatures is only so much, right? I mean, you know, this coral reefs only extend up to about where we are on the east coast of Florida, and well, up in Bermuda, but otherwise, because of the Gulf Stream, but otherwise, you know, on the eastern seaboard, they kind of stop right around here, these tropical reef-building corals. So their temperature range of the corals themselves that they can tolerate is you know, not so great. They do die if they get too warm or too cold. But the disease did spread through the Keys in the winter and summer. We couldn't really see a lot of pattern. In the Keys, one thing they've noticed down there is that in the summer months when the coral starts to bleach, they don't see as much disease, and we don't really understand that very well. We do know that the disease is affecting the zooxanthellae. The people who've looked at this disease microscopically, the histologists, have documented that the zooxanthellae start to die and break up in the disease. So it is affecting them. We don't know if that's, you know, if there's a pathogen, you know, some kind of bacteria or virus or something that's directly affecting the microalgae, or if they're reacting to something the coral's producing because it's sick, or some pathogens or something are producing. We really just don't know, but you can see those zooxanthellae in microscope slides start to almost disintegrate, if you will. So temperature is important in that regard. It may actually slow the disease in the summer. But it also, warm water temperatures can stress corals, right? And I talked a little bit about the difference between this disease and bleaching at the beginning. And we know that one consequence of water temperatures that are too warm for corals is they bleach. And corals that have bleached are compromised. You know, that's not a healthy thing for them. And sometimes are more vulnerable to or more susceptible to diseases. So there's a lot of interconnections between water temperature, but certainly in Florida, it moved around in both the summer and winter months.
Okay, all right. We have another question from Arnaud. I have probably mispronounced your name, so I apologize for that. They ask, those bacteria from diseased coral, when you isolated them, did you analyze the genome to determine the genus and the species? And if yes, did you find a dominant bacteria in those samples?
So the microbiomes, the natural microbial communities shift in those diseased corals. But the problem was we don't know if those bacteria that are occurring in the disease lesions are a primary cause of the disease or just secondary infection, right? Like, you know, if you get the flu, you can get pneumonia, right? And that's a secondary infection because of the weakened state of the host itself, you know, in the case of the flu of humans. But yeah, we just don't know. We do see microbial shifts in those, and that some of that work's published in that paper by Julie Meyer I mentioned that's in Frontiers of Microbiology, and some other research groups have been looking at that as well. Everyone consistently sees shifts, but some of those bacteria that show up in the disease lesions are showing up in sick corals in other parts of the world even, you know, so not related necessarily to stony coral tissue loss disease, but other diseases as well. So they might just be taking advantage of, you know, dying coral tissue and susceptible hosts. So trying to figure out the primary cause has been challenging.
Okay, yeah. Lewis asks, is it thought that the treatment with probiotics would be an ongoing effort, or would the corals be colonized and then not need ongoing treatment after that?
Yeah, that's an excellent question, and one that we've been, you know, well, that's one reason of trying to do these field trials, right, is to actually see what happens. Our hope was that they would get colonized, right, and that this microbe, which was originally isolated from some cavernosa, that the way we had isolated it was we were doing these experimental trials where we were, you know, testing transmission of from diseased corals to healthy corals. We had a couple fragments that never got diseased, and we're like, oh wow, these guys are kind of special. Let's look at their microbiome. Maybe they're producing something that is protecting them, and those were actually the source of MCH17. MC stands for Montasteria cavernosa, of course, and H stands for healthy, because the coral stayed healthy even after being exposed to disease. So, you know, our hope was that they would colonize other Montasteria cavernosa and not need repeated treatment, because clearly to make this practical, to really treat corals in the field, I mean, you can't go out every couple of days treating corals, right? You'd want it to last at least months. Ideally, you know, one treatment and you're done. But so far, we're not sure we're going to get to the one treatment and we're done. But that's why we're trying all these different treatment methods, right? Like the pastes and the application to the whole coral and all this sort of stuff.
Okay, Charlie has an interesting question as well. He's asking, when the coral is diseased, have you guys been observing less aquatic animals nearby? Do the number of animals seem to have anything to do with it?
You know, that's a really good question. We have not done enough of those studies to see, for instance, like, are the fish communities changing and other things. They are starting to look at that in other parts. I mean, well, in Florida as well as in Mexico. Some researchers there, you know, along the Yucatan there, I showed you how it had spread in less than a year along the entire Mexican coastline there of the Yucatan. They're starting to do more studies looking at how the whole community has changed. The earliest papers have all looked at how the coral communities have changed, and of course they've changed a lot. We've lost a lot of corals to this disease, and so these species that are the most susceptible, like the pillar coral, Dendrogyra, is very, very hard to find in the wild in Florida now. I'm not even sure anyone has recently. So yes, it's definitely impacted the really susceptible species. But I do know about other things like fish and everything else.
Okay, sure. Rusty had a couple of questions. He's coming to us from Houston today, and he asks, has the disease been observed at the Flower Gardens National Marine Sanctuary in Texas? And does it seem to be related at all to white band disease?
So white band disease, well, let me just first answer the Flower Gardens. I do not believe it has been observed there. I have not heard that it is or has been. So that's a good thing. White band is another tissue loss disease. So it's, you know, it's similar in that the coral just sort of dies away. But it affects Acropora species in the Caribbean. So that the Acropora cervicornis, the staghorn coral, and Acropora palmata, the elkhorn coral, were devastated in the 80s and 90s by white band disease. Their population numbers have dropped a lot. So interestingly, the white band, as we know it, does not affect these boulder corals that are susceptible to stony coral tissue loss disease. Stony coral tissue loss disease, thankfully, does not seem to affect the Acropora species. So we've got, you know, kind of two diseases affecting different kinds of corals. And together, unfortunately, they're almost all impacted. So, you know, you certainly don't want both outbreaks going on simultaneously. That would be very bad. But yeah, white band disease has also been very devastating. And similarly, even after it's been studied for decades, still don't know what exactly causes it. You know, we do for sure know that corals that are stressed by all this bleaching and temperature and, you know, poor water quality and everything can be more susceptible to disease. And so there's certainly, you know, implications that these corals have just been kind of stressed out a lot.
Okay. Well, this next question I think is sort of apt in this age of COVID. Nana asks, could some sort of vaccination be possible for this disease?
Yeah, I mean, it could. The problem, you know, the difference here is COVID, we know what causes it, right? It's the SARS-CoV-2 virus or whatever. It's been isolated. It can be grown in the laboratory. It can be studied and studied and studied. I mean, thankfully, the science community has found out so much about this virus over the last year. And that's how a vaccine was targeted and developed, right? They knew exactly about these spike proteins on its surface, and the vaccine can target that and everything. So the difference here is we don't know what's causing stony coral tissue loss disease. So perhaps if we can figure out what is, then vaccination is possible, because certainly vaccination has been used for other wildlife diseases. It's not easy. I mean, sometimes they can put it in the animal's food and that sort of thing, but.
Right. Yeah. And like you said, it's not like you can go out there, you know, every few days if it needs boosters and things like that. It's just not real practical. So, okay, well, I think that's going to be about it for us today. If we did not have time to answer your questions, Jamil will be putting our email address in the chat box. You're welcome to email us. Our email address is smseducation. And again, please feel free to register for our upcoming Marine Science in the Morning programs. Our next one will be on February 10th, and we'll be talking to Catherine Harris of the Belle Isle Conservancy Foundation. She'll be talking about maintaining live model ecosystems similar to the ones that we have at the St. Lucie County Aquarium. Again, these are all free programs. They will be archived on our social media. So if you don't have a chance to join live, make sure to check it out on our YouTube page. Val, thank you so much for joining us this morning. I really appreciate it. We got some really good questions, and I think our audience really enjoyed hearing about it as well. So thank you.
Yeah, thank you. I'm sorry, it's not the happiest topic, but fortunately there's some treatments are being developed, and
Right.
I've been working super hard on this and very, very collaboratively and collegially in Florida and elsewhere. So it's definitely, there's some hope here, but
yeah, absolutely. I think sometimes that's the best you can do. We're looking at it, there is hope, and we'll see what we can do in the future. So,
yeah.
All right. Well, thank you so much for joining us, Val. Thank you, everybody, for joining us as well. Jamil has posted links to our Facebook page and our YouTube page. Make sure to check out all of our content there. We also have virtual field trip options for any educators out there, whether you're a homeschool educator or a classroom educator. Those are free materials. There's distance learning materials on our webpage as well. So thank you so much for joining us, and we'll see you next time.
Bye, everyone. Thanks.